So, dear colleagues, while others are joining, let me welcome you at today's conference on research infrastructure. It's organized by the network Environment and Health. That is a network of academic organizations that have been collaborating in the environment and health nexus for last 12 years. I have send you in the chat a link to our website, so please have a look there, and in case you're interested in joining this network, let me know. Also, we are organizing this conference on behalf of partnership PARC that is focused on chemical risk assessment, specifically work package 9 of PARC that is dealing with building research infrastructure in chemical risk assessment. As I said, we are recording this meeting, so I hope everybody is fine with this. And last thing, in case you have any questions to the presentations that are projected on the screen, we'll go without break. So please put your questions in the chat so that we can, after the presentations are finished, we can deal with them and answer them. So without further ado, I'll give the floor to my colleague Shachar, who will start with the presentation on the introduction to ESFRI. Shachar, please.
Hi.
Maybe a few words before Shachar, I'll send you the presentation. And sure. Just to explain, since we are dealing with the research infrastructures, we decided to start at the beginning with the intro of what the research infrastructures are and what is the European Strategic Forum for Research Infrastructures, because some of you may not be familiar. And then we will go to specifically to EIRENE, infrastructure on environmental exposures. And then we will show some examples of the Czech national node of EIRENE. So Shachar, go ahead, please.
Yeah, so you can switch to the next slide, please. Yeah, so what is ESFRI? The ESFRI is the European Strategy Forum on Research Infrastructure. It's a body that was established in 2002 to support the develop research infrastructure across Europe. Its stated mission is to support a coherent and strategy-led approach to policymaking on research infrastructures in Europe and to facilitate multilateral initiatives leading to better use and development of research infrastructures at EU and international level. And its goal is to improve access. Yeah, we can a little bit of. I'm not going to cover everything because of the short time, but it's self-regulated. It operates openly and makes decisions by consensus. The important part is that it has delegates from each country and also from the European Commission. The delegates are usually senior science policy officials, which represent their countries' research ministries. And from the EC, it's also a senior official. And the European Commission provides the ESFRI the secretariat and to support its operations. We can go to the next slide, please. So the ESFRI has two key strategic activities, which are the landscape analysis and the roadmap, which also includes monitoring. Next slide, please. And I will discuss each one in more detail. So the landscape analysis focuses on mapping and evaluating existing RIs to inform decision-making. And the roadmap provides a forward-looking strategy that prioritizes specific new and existing RIs for future development and funding. And the roadmap also includes monitoring, which means evaluating the progress of ESFRI projects and ESFRI landmarks. So it's important just to say that within ESFRI, it's divided into ESFRI projects and landmarks. The projects are new RIs in the preparatory phase, the same as EIRENE, and the landmarks are already well-established research infrastructures. For example, you probably know ACTRIS and ICOS. In the landscape analysis, it serves as a foundation for understanding the current state of research infrastructures, and the roadmap builds on this knowledge to plan for the future of Europe's scientific research infrastructure. So next slide, please. So the landscape analysis, so a little bit about the purpose and the focus. So it provides a comprehensive overview of existing research infrastructures across Europe within specific domains. So there are six domains, which you can see on the right side. And EIRENE, for example, belongs to the health and food. It aims to map the current landscape of RIs to identify strengths, gaps, and opportunities for improvements. And it also helps stakeholders. Okay, so next one. Here we have an example of how the domains work. So we said there are 6 domains. This is the environmental domain, which includes 8 landmarks and 3 projects. And here you can see kind of the distribution and also the synergies, not just within the environmental domains, but also within the health and food. The health and food here is in orange, and the environment is in green. So you can see EIRENE, for example, how it synergizes with other research infrastructures. Next slide, please. So the content, what's in the landscape analysis, it's a detailed snapshot of the current RI landscape. It categorizes by, scientific fields, like we said, the different domains. It identifies synergies, potential duplications, and gaps in infrastructure capabilities across Europe. And it assesses the functionality, relevance, and so forth. In terms of the process and the frequency, it was first introduced in 2016, and the findings inform the roadmap. So it's important to remember the two of them are connected to each other, but now they are done separately. Next slide, please. So audience is of course policymakers. It helps countries and institutions identify existing RIs they can leverage or collaborate with. And the impact is to provide a strategic overview that informs the roadmap, like I said before. Next, please. Yeah, so this is just a summary. You can go over it later, but we don't need to get into it right now. So next, please. So the roadmap, this is the other part. So the purpose and focus is it outlines S3's strategic vision and priorities for developing new high-impact pan-European RIs or upgrading existing ones. And it includes projects that S3 considers essential to advancing Europe's scientific capabilities over the next decade. So it's kind of a long-term approach. And the content, it presents a list of prioritized new and upgraded research infrastructure that are deemed critical to Europe's scientific infrastructure. So like I described before, it's divided into projects and landmarks. Next slide, please. Yeah, so the S3 uses what's called a life cycle approach. It's a six-stage approach from concept development all the way to termination of research infrastructures. EIRENE, for example, you can see on the drawing on the right, you can see where EIRENE is sitting. So we are now in the preparatory phase. And also this is important because within Horizon Europe, there are different calls related to different stages of development. Next one, please. Yeah, so updates, the roadmap updates every 4 years. And the next one will be in 2026. Yeah, next, please. Yeah, next slide, please. Yeah, so the impact, so it acts as a strategic investment guide for developing the most impactful research infrastructure in Europe and often leading to funding decisions and collaborative projects. Next slide, please. So the roadmap, the new projects are being evaluated, and there are different criteria. This is what EIRENE had to go through several years ago. EIRENE was included in the roadmap. It will be discussed later by Anna, that in 2021. So it has to kind of through the evaluation, so to show us a scientific case and an implementation case. And of course, a very important thing is it needs to get support, political and financial support from the country, because the countries eventually, the member states are the ones that are members in the different research infrastructures. Next, please. Yeah, next one. I won't. This is too much detail for now. And monitoring, that's also very important. Monitoring is for research infrastructures that are already on the roadmap. So IRENE, for example, was included in the roadmap in 2021. So it goes through monitoring every several years, and this is also true for landmarks. The way that it works in ESFRI is you have 10 years to reach the operational phase since the time that research infrastructure is included in the on the roadmap. So this is important. Next, please. Next slide, please. Yeah, so this is just this covers pretty much what ESFRI does. I just wanted to mention a little bit the legal models of ESFRI RIs because they're kind of different legal models. There is national legal model entities, for example, like CERN, which operates under the Swiss law. There's public-private partnerships, such as the European Spallation Source. There's intergovernmental organization like Elixir, but the most common one is the European Research Infrastructure Consortium, and probably Anna will mention it a bit more later, discussing IRENE, because this is also planned. This is the legal model that we plan to have for IRENE. So in any case, it's the most typical one. Currently, there are 28 ERICs, and it's an EU legal entity, and it's recognized across all EU member states. It's created through decision of the European Commission. It has a non-profit focus, and it has tax exemption. So there's a lot of advantages to it. So with this, I will finish. Thank you.
Thank you very much, Shachar. So I'm sure that for those of you who are not familiar with the ESFRI concept, this was quite overwhelming, but we really wanted to start with the overall information about what European Strategic Forum for Research Infrastructure is, that it is really the intergovernmental organization that coordinates the development of the large research infrastructures of the pan-European interest. So we believe that this is also what exposure science needs, or the chemical risk assessment. And this is why already since 2017, we were building the community that would help to develop the pan-European infrastructure on the exposure assessment. And in 2017, we sent the first applications for the ESFRI. And we helped to identify the gap, because as Shachar explained, the first step is that the gap in the current roadmap needs to be identified in the landscape analysis, and only then somebody can submit the application for the European infrastructure. So this is just a citation from 2018 ESFRI roadmap, where it was identified that we don't have anything that would look into the environmental exposures, chemical exposures, also nothing on the longitudinal cohorts and the exposome research as such. So this helped us to open the door for the new application for the new research infrastructure. Also, I hope you understood from Shachar's application that this is not a research project, that this is the application that needs to be submitted by the country. So the ministry that is responsible for research in every country can submit the application, and such application needs to be supported by at least 3 other countries in order to be considered and evaluated. So we as the scientists can develop the scientific concept, but in the end, it is a highly political thing, because it is a combination of the European and national funding. So in 2020, we prepared a full application for new EIRENE research infrastructure on environmental exposures. It was submitted by the Czech Republic, supported politically by 6 or 7 other countries. You can see on the left side the citation from 2021 S3 roadmap, where EIRENE was for the first time included as the priority project. And you can also see the current situation. So those are the countries that are currently involved. Those on the first line are the countries where they have not only political support, but also they are included in the national roadmap and have certain financial support. Then on the second row, there are countries with the political support, but no financial support yet, and so on. The last 4 countries are the countries that just joined the consortium later. You should understand that this is the open process. So it means that if your country is not yet included, if you are interested, you can just contact us and be included. It doesn't have any financial implications yet because we are in the preparatory phase. We are still working on the whole concept, how the infrastructure will look like in the future. If you are from the country that is listed here, but you are not part of EIRENE yet, you can contact us again and we will let you know which institution is the national coordinator now. So this is just the overall concept. Again, you've seen this in Shahar's presentation that development of such European infrastructure as the new legal entity takes time. It typically takes 10 years. So we are right now in the preparatory phase. So we are preparing all the legal documents, the content, description of the services. And then next year, we want to move to the next phase where we will try to establish the new European Research Infrastructure Consortium as the European legal entity and set up the services that will be offered to the scientific community. Of course, we are building on the previous development in the field of environmental exposures and biomonitoring project, but also a lot of human exposome projects. You know that the exposome projects were financed by already Framework Program 7, then Horizon 2020, and now also Horizon Europe. But you should be aware that the parallel activities are happening in the US. They started with the Exposome Center already back in 2013 when they started financing the Hercules Center. And now there are parallel activities in Europe and in the United States. And you probably noticed in my previous slide that we also have the US and UK partners. So it means EIRENE is not only the European Union centric. I mentioned that there are human exposome projects that were paid from Horizon 2020 and Horizon Europe. Under Horizon Europe, there is EIHEN, International Human Exposome Network project, that is supposed to build a global exposome network. So it means we are working together with the partners in the US, but also Canada, South America, Africa, Asia, and many other continents to develop the joint concept of how the human exposome research should look like. So it means this is only a CSA action. It's not the research project, but it provides us with the platform to communicate across the continents and develop the harmonized approach, because harmonization is the key thing for development of the research infrastructure, because we have to make sure that we will be serving the whole international community. Of course, EIHEN is not the only pillar we are building on. Many of you are part of PARC, the European Partnership on Chemical Risk Assessment. And PARC project was developed on the previous joint program, Human Biomonitoring for Europe, that was focused on developing the capacities for human biomonitoring. And already during the HBM4EU project, we have noticed that we are building something that should survive beyond the HBM4EU project. So when we were developing the concept of the PARC partnership, we made sure that out of 9 work packages that are shown in this picture, we also have 2 work packages that are dealing with the research infrastructures. In PARC, it is work package 9, which is looking on development of both infrastructural and human capacities for future chemical risk assessment, but also work package 7 that is looking on the data resources. So it assesses existing data resources, their fairness. We are working towards verification of those resources, but in both those work packages, we are also analyzing the best options for sustainability of all those infrastructures. And EIRENE should help to sustain the capacities developed not only in PARC, but also in EHEN and other relevant European projects. I said that there is parallel activity going on in the US because National Institute of Environmental Health, together with 2 or 3 other NIH institutes, are now financing the new Nexus project, which is the network for exposomics in the United States that has a very similar mission. In fact, it is a combination of the European EIHEN and European EIRENE, so it has both the infrastructural part and networking part. It is coordinated by Columbia University in New York, and they want to harmonize the whole exposomic field in the US, but also serve as a partner for Europe and beyond. And of course, we need to be aware that we are not alone in the ESFRI landscape because there is also many other relevant infrastructures, both in the environmental domain and human health domain. So it means there are infrastructures like BBMRI looking into the biobanks, ECRIN in clinical studies, EATRIS in translational medical research, but also the ones focused on biology, developing models of diseases, the microbial communities, pathogenic agents, and so on. In the environmental domain, we have infrastructures looking into quality of air or water or ecosystem biodiversity. So it is very important for us to be in touch with all of those other infrastructures to make sure that we will leverage the capacities that are already there without duplicating things, and that we will only build things that are necessary. I should mention especially the infrastructure Elixir that is providing data and computing resources for the biological and biomedical research. And we are also closely working with Elixir and want to use the capacities of the Elixir infrastructure, but also the EMBL, the European Molecular Biology Laboratories, especially their center in the UK, who is providing the computational capacities for whole Europe. So speaking a little bit about the structure of EIRENE, how it is evolving now, we believe that EIRENE should provide services that are clustered around 6 pillars. First 3 pillars, here shown as ABC, are focused on the laboratory capacities. So it means providing an access to the chemical laboratories that can give the information about the exposure biomarkers, laboratories that can measure effect biomarkers. So it's a combination of the laboratories that are based on sequencing analysis and mass spec analysis, but also toxicological laboratories. While D, E, and F are dealing more with the data. D is an access to the environmental data, E, an access to the human data. But here, it's not only access to the data, but also access to the samples in the biobanks of the longitudinal studies. If such studies have the samples, but they didn't measure the exposures, and they can provide the samples that we also need to have this information. And under F, we can hide any tools that are dealing with the data, so risk assessment models, pharmacokinetic models, AOPs, and all the other tools or models that are dealing with such data. And then, of course, what is key is that we need to have some internal services, and it is all about harmonization and standardization of all approaches, because we have to make sure that if we want to develop the analytical capacities in Europe that is capable of analyzing hundreds of thousands and maybe millions of samples, we need to work the same standard operational procedures and work along the same quality assurance standards. So, of course, part of the central services would be quality assessment of both laboratory facilities and data. And here, those of you who are part of the PARC understand that this is what work packages 7 and 9 are about: building the QA/QC system for the labs, for the data, building the inventories of existing resources and really actively working with them. But we also need to very actively work with the potential users. So we need to develop the network of the users and communicate with them on the future needs of the research infrastructures, because the research infrastructures at the European level are financed under the excellent science, under the pillar 1 of Horizon Europe, which means that this is not the routine service facility. It's really the mechanism that should develop technologies, services, and human capacities that can address the questions of the tomorrow, not just today. So one thing is building the internal system and harmonization, collaboration with other infrastructures, but then we also have to think about who will provide those services. So currently we are discussing which facilities will be included in the pilot service providing, and in which of our national nodes we already have the facilities that are matured enough, that have the financial support and have the capacities to provide the services to the international projects or the institutions asking for such services. I should mention that for the whole development of the concept is important compliance with European open science. So we always think about sharing whatever we have, be it standard operational procedures and libraries and data resources, making sure that we have the proper system of the metadata and that all data will be findable and reusable in the future. And we are especially making sure that we leverage all the existing resources. And I already mentioned the collaboration with Elixir, EMBL, EBI, and so on. The research infrastructure, once it is established as the ERIC, is expected to provide the services for 1-2 decades before it gets upgraded. So it means that we have to think about long-term sustainability. And this is why the collaboration with the research of the research infrastructure should be established with all the existing European projects. And European Commission is really considering that in the future FP10 projects, they will really demand that the collaborative research projects always collaborate with the research infrastructures to make sure to use the technologies and methodologies that are state-of-the-art and to make sure that all the data generated and stored within the project will be preserved for the future. So that much about the EIRENE infrastructure, and since this may feel a little bit vague, I also wanted to show you how such research infrastructure looks like in reality. And the reason why RECETOX Research Center coordinates the European EIRENE infrastructure is that RECETOX has been serving as a national research infrastructure more than 10 years already. So it has been part of the national roadmap since 2011. So the whole RECETOX center is the environmental and health center operating along this triangle. So it means connecting the environmental research with the research on human health and well-being, but also a lot of research on sustainable societies and the living laboratories and so on. And as I said already in 2011, we figured out that if we really want to build longitudinal networks or cohorts or laboratories that are sustainable, we need to develop it as the open access research infrastructure. So since the beginning, we have several core facilities. We are operating the monitoring networks that are supporting not only all European countries, but also some countries in Africa and South America. We have several in-house population cohorts. We have quite extensive accredited laboratories, and it's all required that we also developed internal information system, including environmental information system, Genesis. And in the last 5 or 7 years, we are also increasing the capacities of our laboratories to have also the laboratories of the biomarkers, looking into the metagenome, genome, and so on. And because I wanted to give you an idea of what kind of services can be offered through the capacities of the research infrastructure, I invited the heads of our research facilities to tell you more about our core facility units and the services that are providing. So we will start with our trace analytical laboratories, and I would invite Petra Přibylová, who is the head of the trace laboratories, to say a few words about them. Petra, please.
Thanks, Jana. Good afternoon. My name is Petra Přibylová. I'm originally working in RECETOX in research infrastructure that I would like to show you activities of core facility of central laboratories. It is since last 14 years busiest part of RECETOX research infrastructure. Since last 10 years, we are accredited according European standards for environmental sampling and chemical analysis. Since last 6 years, not only for environment, but also for human samples. Accredited laboratories provide analytical background for the research programs of all RECETOX Centra and provide analytical advanced services to our customers from RECETOX and also for external clients. We are responsible for analysis and long-term monitoring programs, like Jana already mentioned. Next laboratory that we are focused on is microbiome laboratories. It's quite a new core facility, provide the bacterial and microbial profiles in various samples, mainly using 16S RNA gene and whole metagenome sequencing. Another part of laboratory is biomarker analysis laboratory that Elliot will introduce you later on. So the services of trace analytical laboratories is development of samplers for non-polar and polar compounds contamination, and develop analytical methods for selected chemicals, pollutants, and their metabolites in human exposure studies. We are able to make such service on each customer individual request. So next slide, please. So since sampling, we are able to offer to make sampling plan, to organize trainings of sampling, or to use our technicians for sampling campaign in Czech Republic and abroad. We are working on sample preparation, analysis, data evaluation. From the position of accredited lab, it's necessary to follow QA/QC parameters and to establish and follow QA/QC system in each analytical method, and we are responsible for long-term monitoring studies. Next slide. Compounds of interest. Mainly it's about organic pollutants, persistent ones and emergence in last several years. its compounds belong to Stockholm Convention, metals on Minamata Convention, and for human exposition, the most important are polar compounds, like, for example, perfluorinated compounds, bisphenols, plasticides, thyroid hormones. Now newly we have added UV filters like benzophenone, mycotoxins, and metabolites of such native compounds. We also make method development, so now the focus is mainly done in species analysis in human samples, and we have all the time methods for development. So next slide. In the sampling services in environment, we have started in outdoor environment, mainly with air. We are responsible more than 30 years for active air sampling and provide the data to, for example, EMAP, EBUS database. Then we have moved to passive air sampling that was developed with our colleagues from Lancaster University and Environment Canada. We are responsible for monitoring networks and sampling of water, also active and passive methods. We have capacity to work on sediment, soil, biota sampling, and concerning atmosphere, also atmospheric deposition. Next slide. So, like I already said, we have accredited laboratories under international ISO norm. Also in accreditation process, we have started with outdoor environment matrices, but then we have moved for human matrices. So now we have under accreditation also metabolites or metals and species. Next slide. Active air monitoring activities we are follow since 80s in last century, mainly for pHs and, for example, for persistent compounds like PCBs, OCPs. We are offering this data to European database EBUS under EMAP program. Next slide. And finally, I would like to introduce you microbiome laboratory. That is quite a new facility of RECETOX research infrastructure that analyze bacterial profile using modernizing methods. We are working with human samples, especially with stools, buccal swabs, meconia, and also we have several studies of environmental samples connected to microbiome. It is about dust and soil microbiome, also for water. And the method, procedure are DNA isolation and preparation of library, sequencing, and such kind of procedures. We have mainly two methods, 16S RNA gene sequencing, DNA isolation, and now newly whole metagenome sequencing. So it's maybe all from microbiome and trace analytical labs, and now my colleague Elliot will continue with introduction of biomarker analytical labs. So thanks a lot.
Elliot, the floor is yours.
Hi, thanks. So essentially, as Jana previously mentioned, biomarker analytical lab was established relatively recently. We have a suite of instruments that encompass GC Orbitrap, LC Orbitrap, and also triple quad LC technology coupled to analytical and low flow systems. So the next slide. So essentially, we cover three domains. So one is to do quantitative assays of proteins, including their variants. and then multiplex metabolite assays, which are again quantitative for endogenous analyte. These two then essentially complement the trace analytical labs of Petra. And then also, due to the technology, we then do profiling assays, so full scan exploratory analysis and screening for both chemical agents and endogenous analytes. So essentially, one of the core aspects is the fact that we acquire a lot of raw data along with all metadata and upstream and downstream processes, but basically the auxiliary information that is typically used to actually reproducibly process this complex data is often missing. And we just push the, in addition to making the experimental data open, the auxiliary information should also be as open as possible for reproduction, because this is where the field in the last 20 years has not really been replicable on the full scan analysis. And the next slide. So essentially, if you don't document the approach, it isn't reproduced. And within these domains, there's actually few labs that share the SAPs, the resources, pipelines and processing, along with how they do QA/QC. And typically, not many labs within these domains have accreditation and also the procedures for reporting full scan assays. And for example, regulatory work, there are not particularly well developed frameworks. So typically, SAP sharing also has two approaches. The top side is then from one of the largest US labs that was funded under the Common Fund and is essentially an NIH infrastructure lab, where they have an SAP that essentially has 30 parameters, and then our comparable SAP has around 200 parameters, along with them 5 accompanying SAPs for all steps, which then just evidences the amount of work and effort gone in to share as much information as possible for replication, which will be needed in multi-site standardizations. Next slide. So just as an example, essentially on protein quantitative assays, this is just because the majority of work in proteomics is again using full scan. It's relatively standardized, but in proteomics it differs to the small molecules, whereby actual absolute quantitation is relatively inaffordable. And this is largely due to the fact that they need to label entire proteins due just to the digestion and enzymatic treatment during sample prep. However, within our infrastructure, we essentially utilize what are called winged peptides. These have been compared, they're winged on both the C and N terminus. They've been compared to fully labeled protein and essentially give accurate but more affordable results. So they're basically comparable in their data accuracy, but are far, far cheaper, which then means that we can multiplex assays for analysis of more proteins, including their isoforms. So at the moment, we just have a panel for blood and fecal swabs at the moment where we're looking at immunity and inflammation markers, just because it functions into the hallmarks of health. And then we're going to be extending this to intracellular signaling and also hopefully neuroproteins to hit developmental stages. And then the next slide. So essentially, at the moment, when we deliver our data, basically the raw data along with the actual pre-processing is fully open. So we deposit it into a public web archive, which means that essentially everything is anonymized, but we can deposit the raw data files and then they are given a permanent URL. And it means it's also accessible. So this is just an example of that. It then had all information about which peptides were measured, but it also contains the actual peak integration, for example, the calibration curves, the QA/QC data, etc., so that anyone can actually check, 1, the validity of the measurement, and 2, they could essentially interpret or leverage that data and couple it with their other studies. So it just fits FAIR principles for reuse. On the metabolite, we have various panels. We specifically focus on blood, where at the moment we just have panels for short-chain fatty acids, the tryptophan pathway, and fat-soluble vitamins. On the right-hand side, it's where, unlike other methods doing broad quantification, we specifically chose relatively few metabolites per panel to keep costs down, because each one then requires the respective label standards. So we had a minimal number of analytes as possible to encompass as wide a range of cellular processes, which are evidenced by the colors on the right-hand map. And then also underway, we're working on extending this specifically to redox metabolism, and also the Barclays kit for fall automation and analysis, just because the Barclays kit have heavily been used in many large-scale cohort studies, and some of the kits already allow multi-instrumental model application and interoperable data to be generated for core metabolism. And fully automating makes that again more affordable. On the non-target or full scan analysis, essentially at the moment for the LC work, this is a broad field where not many labs have aligned work, and it's actually quite difficult to harmonize LC work. There's a deficit in heavily curated libraries that encompass retention information. So we also generate our own spectral libraries that then actually include the retention information from the chromatography along with substructure annotations. These libraries are fully available, and then basically for our methods, we propose that full characterization of the method, where we run thousands of standards on an actual system and then characterize the chromatography. We also release the models and the accuracy of those models to predict retention, which is then needed for suspect screening and annotation of analytes where commercially available chemicals are not in existence, or also where the concentration of analytes is not high enough to actually allow other structural elucidation, such as NMR, in a feasible manner. So essentially, it's just to increase the confidence in annotation and report the QA/QC of how we've done that. And this is used, and we contribute to various projects with this work. And then the next slide. And then also on the GC side, largely because fewer labs do GC, it's much easier then to test full standardization. Also because just due to the instrumentation we purchased, we have robotic auto sampling directly on top of our GC instruments, which then meant that they have various modules, but we can do all procedures such as centrifugation, micro SPE, vortexing, etc., in a robotic way, which then obviously takes away individual error, which then meant that it's quite easy to standardize. So essentially at the moment, we just have a small group that also encompasses labs within EIRENE, such as Leiden, Vienna, and also labs that cover both EIRENE and then the US Nexus project of Columbia and Emory, and this falls then within the EHEN project. But basically, we've got a multi-site replication already of a method which we established here, that in the next slides you will see is already being applied to multiple cohorts within the European Human Exposome Network. So here is just example data that was from a small pilot study focused on the Helix cohort within the Athlete project, where it was around 800 samples, and we're currently working on a manuscript for this data. And then on the next slide, we've also applied this to multiple studies within Expanse, encompassing nearly 7,000 samples, and we have an upcoming project then also upscaling this to tens of thousands of samples, and essentially the QA/QC is shared, and then the raw data and everything will be under publication. And then in cooperation with PARC, we're inventorizing the FAR assessment of the mass spec data banks, and also working in a different project to actually develop then the EU repository for MS data, just because there was a lag in funding and in development, which didn't extrapolate that repository from endogenous to then also be amenable to exogenous chemicals and also associated environmental metadata. And that just lists. Well, then I guess I'll pass over to Helga on the data side.
There you go. thank you. So accompanying the analytical labs, we're also developing software and data processing pipelines. And similar to the analytical laboratories, there's a big focus on reproducibility, robustness, transparency, and general like high quality tools and workflows. So all source code for the software tools is developed open source and is continuously tested and deployed using GitHub Actions infrastructure. We develop elaborate test suites to really validate that the software is bug-free and works as it's supposed to. And this also includes static code analysis tools and code coverage to see, for example, where there are downsides in certain parts of the code that might not be covered by unit tests, etc. All of the software is then distributed directly as installable packages using common repositories such as PyPy or CRAN, or actually binary redistributions like Bioconda and CondaForge for improved reproducibility. Further on, these packages are containerized to actually allow scheduling via cloud computing infrastructure, and we wrap those tools for the Galaxy platform, and we're collaborating a lot with the Galaxy project to make the tools accessible to researchers who don't have a programming background, so that people who don't know how to use command line or write software can actually use these tools, and also to integrate this with like larger data infrastructure. So yeah, the tools are available on most useGalaxy.star instances. So far, we're covering useGalaxy U, the Australian instance, the Czech and the French instance. And furthermore, user trainings are provided by the Galaxy Training Network. The next slide, please. Yeah, so far we have released more than 20 open source code repositories and have contributed more than 60 tools to the Galaxy ecosystem. We are maintaining more than 20 software packages, so this means packages on Bioconda, PyPy, CRAN, etc. These tools are combined into two end-to-end workflows, which are registered on Workflow Hub, and from there they get assigned a DOI and they can be directly executed on Galaxy instances which provide all available tools. In this case, this is useGalaxy U. Trainings for those workflows are also available via the Galaxy Training Network. And as already mentioned, since this is like on the computational side, we are of course involved in Elixir through the communities. So this is specifically the metabolomics community. And we're also engaged in further community building activities regarding mass spectrometry data processing within Galaxy. Next slide, please. And so this covers the upcoming work. So Galaxy itself covers the data processing needs that we have and storage of results, etc. But the vision includes setting up a larger connected data infrastructure that also includes the raw data management. And there, the perspective is to also use well-tested open source software systems such as IRODS for the raw research data management and storage, and to connect this with Galaxy and the Elixir Life Science login to have a unified authorization infrastructure, and within the framework of Elixir to extend existing ontologies and Elixir core interoperability resources to use those for metadata annotations. So to use standardized data formats, procedures, ontologies, etc., to make the data and tools findable and interoperable with other systems. And that will be all regarding the data infrastructure.
Thank you very much, Helge and Elliot. You know, from listening to this, and I'm sure this was again quite overwhelming, you should understand that we are investing a lot into the quality and standardization of both the laboratory procedures and data processing pipelines. And you should understand that this is the major role of research infrastructures. This is exactly why the whole S3 was established, to make sure that we are investing not only into huge technologies like CERN, but especially if we are building the big pan-European networks, they really have to be built under very high quality. So this is why we are also trying to leverage anything that is already there. Helge mentioned a lot of collaborations with Elixir. He also mentioned the Galaxy community, which is actually the community that was born in the United States around the genomic data. They figured out that they need to join the forces to develop the processing pipelines that can deal with the large data. And we are now in the same situation with the non-target screening data. And this is why we are trying to leverage the Galaxy pipelines. And in Europe, Elixir is the infrastructure that took them on board. They developed it already for sequencing data. They are providing us the fantastic support in building also our MS data processing pipeline. So this is where we are always looking for the support and help where there is already money and resources, rather than building something new and asking for the new financial resources. So that much about our laboratory capacities, and I'll give the floor to Lenka, who will speak more on the population cohorts and the samples that are available for the research and for the laboratory analysis. Lenka, please.
Thank you, Jana. Good afternoon, everyone. I would like to present the RECETOX cohort studies and Biobank as a platform for environmental and human health research. Our focus is on understanding how environmental factors affect human health. We built comprehensive dataset and sample collections to support longitudinal and cross-sectional research across different populations. We conduct several population studies, most of them under the acronym CELSPAC. These long-term studies are unique in Eastern or in Central Europe. These studies involve nearly 40,000 participants and follow individuals and families across generations, providing data on exposure factors and how these factors influence human health over the course of a lifetime. Next slide, please. We collect a wide range of data from study participants at repeated time points using online self-reported questionnaires. This includes details about their lives, such as their lifestyle, nutrition and diet, socio-demographic factors, subjective health and other parameters. We also gather information from medical records and clinical examination, and we invited participants to undergo specialized non-clinical assessment like body compositions, MRI, spirometry, and other procedures. Next slide, please. A crucial part of population studies is obtaining and processing biological material from participants. We handle various human biological materials, including venous and cord blood, urine, buccal swab, feces, breast milk, and others. The key step is an aliquoting process, which involves preparing small volume samples from primary samples. Depending on the type of biological matrix, we prepare aliquoting from 250 to 800 microliters, and we use automated system Hamilton, which guarantees consistent and accuracy while reducing human errors during entire sample processing workflow. We also use advanced automated system for DNA and RNA extraction. And before storing, each sample undergoes basic characterization, including biochemical and immunochemical markers like lipids, vitamins, hormones, glucose, and other parameters. And these biomarkers are saved in database as individual variables so these analyses don't need to be repeated. Next slide. Our biobank operates under strict quality assurance and quality control protocols. We are a part of biobanking and biomolecular resources research infrastructure, BPMRI, and the entire history of each aliquot from origin and collection to processing and storage is documented and traceable, ensuring reproducibility and reliability. Automated storage system Ascion are used to minimize human errors and maintain a stable temperature chain for obtain sample integrity. As I mentioned before, each sample is linked to detailed metadata about its origin and history, including biomarkers, as well as data from chemical analysis of pollutants, particularly from our trace laboratory, and such widely available data on exposure factors and health outcomes can be used for collaboration within international projects, which you are all familiar with, I guess. And the CellSPAC platform offers open access, open request. Now it's over 500,000 biological samples, aliquots, questionnaire and medical records data, and other exposure and health outcome information. And these resources support scientific research and publications in high impact journals. CellSPAC platform combines long-term studies with advanced biobanking to better understand the connection between environmental factors and human health. And conclusion, the summary of services provided by CellSPAC platform includes access to database already collected data and biological material samples. Additionally, the CellSPAC platform serves to design and implement new epidemiological studies, including data, samples collection, analysis, and ethical compliance. And we offer laboratory and biobanking services also, like separately services. Yeah, that's all from my side. Thank you.
Thank you very much, Lenka. And the last core facility we would like to introduce are IT and data services. And this is very important for us because this is also the coordination center of EIRENE. So a lot of things that we are presenting here, we are already upscaling to be able to serve the whole EIRENE infrastructure. So Richard, please take the floor.
Hi, good afternoon. So as you can see, my colleagues introduced already quite a lot of stuff. And to operate and run such amount and variable data collection, data acquisition, and other stuff really also requires a support from IT department. Not only purchasing the hardware, but mostly, or from my perspective, it is running and development of the system. So our Department of Data Services is split into two chunks or two branches. One is focused more on software development, database design, database management. It consists of experienced system engineers and developers. And the second part is focused more on data management and data stewardship and related activities. And those teams together somehow operate to give support both to internal and external customers. So we develop, customize, and operation information systems, database systems, and mostly integrate them together. So we teach them or we develop them to enable inter-system communications, because our overarching goal is to integrate data from multiple departments, from multiple systems across different scientific and research domains. Can we go to the next slide? This is a brief diagram as we think about, for example, the infrastructure within RECETOX. So we are covering from the left side systems and data sources up to data warehouses and places for data processing. So on the data sources side, it could be either an environmental monitoring network like MONET or other campaigns, or we are supporting populational studies through data management systems and supporting tools to really acquire and manage large-scale population studies. Then, because the study usually generates some samples, we help with operations of biobanking system and laboratory information management system. Sometimes we purchase those systems from external providers, so there is quite little work with this, and we also usually give support with the hardware resources and the infrastructure behind them. But sometimes it needs a custom development, and that's the time we can help. Then we developed dedicated systems and dedicated repositories for specialized data types or public portals to give broader access and broader visibility, for example, to environmental data through Genesis system. Then we expect, or then we support the data flow further on to ingest those harmonized and well-treated data into data processing pipelines to the floor or on the table of our data analytical teams, and we gather them together in data warehouses, which are dedicated databases which focus on data integration from multiple sources. So can we go next? So one of the first system we offer as a platform is Genesis, as I mentioned. It's a scientific data repository for chemicals or to track chemical occurrence in the environment. And the goal is to really have a comprehensive data inventory and data database of fully harmonized data, which can be compared together and analyzed together to identify long-term trends, baselines of chemical occurrence of individual chemical compounds across multiple environmental compartments. This system consists of public data portal, the database behind the scenes, definitely the support of our team and data managers, which helps and ensures that all data are really comparable. And also we provide data visualization platform, which serves to a general public, to specific stakeholders like municipalities, national governments, and agencies focused on environmental problematics, and also for international treaties like Stockholm Convention in the case of Global Monitoring Plan. Next, please. So based on the Genesis, we built and further developed the GMP data warehouse, and the GMP stands for the Global Monitoring Plan, which is connected to Stockholm Convention on Persistent Organic Pollutants. And we developed this system to support effectiveness evaluation of this activity. So we support the data acquisition from all data providers, which are scattered all around the world. They are organized into multiple, let's say, regions, so-called regional organization groups. And based on procedures and workflow, which is established and described in the guidance document of the Global Monitoring Plan, we have a system which supports such a flow and organize the data imports, data validations, and approval management within a tool we call Data Management Console. And this tool helps really to integrate data from different sources and prepare reliable and comprehensive data set which is further used for regional reports, and later on is used for a global report on persistent organic pollutants based on the methodology of Stockholm Convention. Again, this portal is also equipped with data visualizations, but this is the example from the data management console, which organized data imports into some systematic approach, and we really want to ensure the quality assurance and traceability of all activities. So all imports, all validations, all changes are tracked and recorded in the system, so anybody is sure that data were not somehow treated unnecessary or not according to the standards. So next. This is again example from the data management console, which is and here in detail on one specific data import, which brings together some metadata envelope together with validation of the file itself, which was stored in the database. And then after all data are inserted in the database, they are published and visualized on the publicly available portal, which serves, as I mentioned, to regional organization groups to develop and conduct the regional reports. Next slide. So this is the example from the data visualization tool, one of many examples I could show, but this is quite significant one. This overview we called map of trends, and because the whole global monitoring plan is focused on observing long-term trends, we try to develop a visualization which really shows whether the contamination or the occurrence of specific chemicals across multiple sites is the same, is increasing or decreasing, and that's the way how we visualize it. So this is the most important information from the GMP put into one map and one picture. Lenka Andrijskova previously mentioned and described that we organize quite extensively populational studies, and also to this department we are giving support through a set of dedicated systems and tools. We can cover under umbrella we call cohort management platform, which is a set of tools which help and really enable to manage large populational cohort studies. The first system is CELSPAC admin, which another is CELSPAC admin is focused on management of cohort studies. CLADE IS is focused on electronic data capture questionnaires. Then we have dedicated systems to collect the data from instruments, machines, and examination rooms. And data extractor is internal tool to dig out data from various formats and transform them into tabular format, so we can later on integrate them in the CELSPAC data warehouse. Next slide. The CELSPAC admin system a little bit more in detail, because that's the brain and source of truth about each and every participant within our studies. So the CELSPAC admin system makes sure that participants are correctly enrolled to the study. It manages studies and sub-studies. We make sure that all sensitive information are treated well, that only really authorized personnel can work with such information. And actually this system is the central core for scheduled events and rule-based and time-based distribution of questionnaires, invites to sampling campaigns, and other stuff. Next, please. because other departments or all the departments together generate quite a lot of data. We are building a data catalog which inventories data sets available and generated within our research infrastructure. So here comes in place the data management team, which helps to describe managed and organized data sets with proper metadata according to fair standards and fair principles. And this is also the way how we make data a little bit more visible, for example, through the integration into the GEOS, which stands for Global Earth Observation System of Systems. Next slide, please. Quite important approach is for us building data warehouses. And data warehouse for us is a database which integrates data from multiple sources. It performs significant and crucial transformations to clean up data, to transform data into format that then later on they are quite easy to use for multiple end users. It could be directly business intelligence tools like Power BI or Tableau or something like this. But here at our institute, we usually work with R, Statistica, MATLAB, and other tools. So data warehouses are the tool we use for proper data integration, and team dedicated, or for each data warehouse, there is a dedicated team who really understands the content of the data they manage and they transform, so the end users doesn't have to. That's another principle we try to develop. We provide quite a lot of training and guidance on this. Even though we are not directly developing those data transformation models, we try to maintain the overall platform so other people can use the same tooling and similar approaches in different use cases. Next slide, please. Even though we are not certified according to ISO standards, we try and we do our best to follow those principles. So I'm originally from the department where this was on daily basis quite normal thing. So we keep going in same rails, and we keep compliance with standards based on ISO 27001 on management of sensitive information. We conduct regular audits on our internal infrastructure development and quality assurance procedures. Next slide, please. On the next slide, I would like to mention that one of those projects we are maintaining and developing is the system which gives the entry gate to the research infrastructure itself through the system of request to open access. So this system provides a catalog of services. It manages the request to the infrastructure and also the process of evaluation and approval management of each and individual request. We collect overall in communication with the requester and evaluators, and then people who really make sure that the request is fulfilled. And we are going to further develop the system into more robust. So we will be using it also to data access, and it's gonna be further developed so it can work as an entry point for services offered by EIRENE infrastructure as a distributed research infrastructure. Next slide, please. And as I mentioned, we have quite skilled and involved data management team. So we work both internally and externally. Nowadays, we are quite involved in several international projects. You may know EOSC activities, or definitely you know EOSC activities, as well as PARC. And here we are involved in work groups focused on metadata development and or the development of metadata standards and schemas, ontologies, and other semantic artifacts. And these people are focused and dedicated on data management activities, data stewardship services, and similar related stuff to help our colleagues, scientific teams, and operational departments to make data more organized, more compliant with FAIR data principles. And I think that's pretty much all from my side, and we can continue with other. Thanks.
Thank you very much, Risha. So again, Risha described the details of the open access. So it means the system that allows our users to ask for all those services that were mentioned in previous presentations. So now in the final part, I will give floor to Petra Ružičková, who is our open access manager, to give you the feeling of what kind of users we have and how many users are really asking for our services. So you have an idea how this research infrastructure functions. Petra, please.
Thank you, Jana, and good afternoon, everyone. My name is Petra Ružičková, and I'm coordinator and open access manager of our research infrastructure. And today I will be presenting on the capacity use of our research infrastructure. Our core facilities are open. They are open to researchers from the Czech Republic and abroad, offering a centralized hub of advanced scientific equipment, open access services, shared expertise, and support for training and education. Our mission is to facilitate cutting-edge research by ensuring a transparent, user-friendly process for both national and international users. The research infrastructure website provides a detailed overview of our laboratory capacities and services. Next slide, please. Our open access strategy is designed to accommodate diverse users, including scientists, students, and industry professionals. We offer all three modalities of access, such as physical access, remote access, and virtual access. These options ensure that users can access our resources most conveniently for their needs, whether on-site or remotely. We provide on-site access for hands-on research and training, remote access for sample analysis and access to data, as well as free online access to data and analytical tools. We also actively promote our technologies, services, and opportunities to attract new users. Next slide, please.
Là, depuis une demi-heure.
Access to our research.
J'ai vu.
Access to our research infrastructure is managed through a clear and transparent application process, as you can see on this slide. Interested users must electronically submit an application form through the online system, which was presented by Risha in the previous presentation. It's needed include CV and detailed project description. We have quarterly open access call, and applications are evaluated in 2 stages to ensure both technical feasibility and scientific quality. Then successful applicants are required to comply with our general access terms, submit a final report upon project completion, and complete an evaluation questionnaire. Acknowledging RI in all published outcomes resulting from using our facilities is very important. The results and outcomes of all open access projects are being monitored and reported to our ministry. The next slide, please. In total, 346 open access applications were submitted for the RECETOX RI capacities in the last year, and based on the transparent evaluation procedure, access was granted to 93% of applicants. The figures illustrate our impact. 98 researchers are hosted on-site, and 230 users accessed resources remotely. Further on the slide, you can see the users' distribution across each core facility. We see strong participation from both national and international users, showcasing the reach of our resources. About 30% of the available capacities of our research infrastructure are kept for strategic research, improving the quality of future services. The remaining capacity is available for open access for external and also internal users and commercial use. The commercial service is provided to both the private and public sectors, but is limited to 20% of the available capacity following public support restrictions. In summary, implementing the RI project resulted in 160 scientific results and outputs in the last year, including scientific publications in highly impacted international journals. RECETOX RI also supports students through numerous theses and courses, organizes scientific events and events for the public, and has nearly 20,000 unique users on its online platforms. The RI has supported more than 130 scientific projects in the last year through its open access to its facilities. As I already mentioned, we also collaborate with public and private sector clients to foster innovation and knowledge sharing. Next slide, please. Our International Summer School is a key tool for capacity building held annually since 2005. And it offers intensive 5-day training on specific topics with lectures, lab exercises, and field trips. The key topic changes every year, and more than 1,000 participants from 100 countries have attended this event so far. This year's edition and the upcoming one planned for the next year are organized in collaboration with and supported by the PARC project. So if you are interested, please follow our webpage regularly for more details, and you are cordially welcome to participate. That's very briefly all from my side. Thank you for your attention, and if you have any questions or need further details, please don't hesitate to contact me at, for example, email, and we will discuss anytime.
Thank you very much, Petra. So the research infrastructures are evaluated based on the good science and publications, based on the users that are applying and served, but also they are evaluated based on the social impact. So we are making sure that all the data is being provided to the public bodies. We collaborate with EMAP, with United Nations Economic Commission on support of the Celera-TAP convention. Our monitoring networks are being reported to UNEP and to WHO, and we also collaborate with numerous European projects as the service providers. RECETOX also serves as the regional center of the United Nations Environmental Program for Capacity Building and Technology Transfer, as the WHO collaborating center, and because the Czech Republic doesn't have the Agency for Chemical Risks, via the contract between the Masaryk University and our Ministry of Environment, we are also serving as such agency as the National Center for Toxic Compounds. So this was the overview of the activities of the EIRENE national node in the Czech Republic. And we went into so many details to give you the flavor of what we are hoping the EIRENE will be in the future. As I said, the development of the European research infrastructure is the long-term process. So we are only building our joint understanding how the whole EIRENE infrastructure should look like. We do collaborate with many European projects on identifying both the needs and existing capacities, and developing the understanding between our scientific partners and political representations riding the national and European project. But basically what we have in the Czech Republic, we are hoping can be upscaled. So we should, as EIRENE, provide an access to the big network of the harmonized laboratories and the biobanks, the accessible the information about existing samples and the data in the national systems. And this is why we are also so active in PARC work packages 7 and 9, making sure that we will bring our knowledge there and help to find the sustainable solutions for this research project. So with that, thank you very much for your attention. And in the final part, I would like to give the floor to Eva Govards and Jos Bessems from the Belgium, EIRENE. And I will stop sharing. And if Eva and Jos can share their screen and start with their presentations.
Maybe, Jana, since we have 3 minutes left, there are 2 short questions. I guess, can we answer them?
We can do it now or after you send Eva, whatever.
Let's do it now. The one has already been answered by Dirk, but I think it relates to your first presentation. Claudia Hepner is asking about how will the data part, the D part that you presented in your presentation, and tools interact with the EU Common Data Platform on Chemicals. Dirk wrote an answer, but I was wondering whether you want to add something.
Yeah, as I said, EIRENE already only needs to establish the official relationship with the European agencies. So far, it is under development. So we have it fully developed at a national level, not only in the Czech Republic, but Belgium, Netherlands, and other countries. But this should be the part of the discussion under the PARC sustainability tasks, how we will manage things, whether we can, as the EIRENE infrastructure, take aboard some responsibilities, whether we will develop some long-term collaboration with certain agencies and stuff. So there is no, legal binding yet.
And there's a question, I guess, to Lenka. I don't know, but I don't know if she's still with us here. If effect biomarkers are investigated from CELSPAC cohort in PARC project, but I guess that can be answered separately.
We so far didn't supply any of those into the PARC because we did supply some of our cohorts to HBM4EU, the teenagers and young adults and small children. But in PARC, the alignment studies are really looking into the national biomonitoring program. So for the Czech Republic, the National Health Institute is offering their samples from the monitoring programs, and the colleagues from the relevant work package were not interested in it. But we are providing our data to other work packages. We are in contact with work package 6, willing to give the data on the biomarkers that we measured to MC-Array platform to be further used for the analysis. We are also participating in many activities of work package 4. So if there is an interest in using the data from our first cohort, young adult cohort, aging cohort, small kids, we really have a lot to offer.
Yeah, thanks. So starting on sharp with Jos's presentation. Jos, can you share your presentation or shall I do that?
I will share it. Thank you
very
much,
yeah.
Lucas and Jana.
Thanks.
Do you see my screen now?
Yes, if you could put it in presentation mode.
Okay.
Perfect.
So thank you.
Yeah.
So I'm Jos Bessems, working at the Flemish Institute for Technological Research. It's a state-owned company owned by the Flemish government, and we are happy to be able to contribute to this conference on research infrastructures. So we have in Flanders now a project where we try to contribute to the IRENE initiative in Europe, to the exposome research infrastructure. And this is a project that has been financed by the government of Flanders. So up to now, we call ourselves IRENE Flanders, but in the end, it's of course the goal to, let's say, extend this to be IRENE Belgium in the next few years, when IRENE PPP will be converted into the IRENE implementation phase, and then finally, hopefully, into a fully European research infrastructure consortium. So just to mention briefly, you know, probably most of you, that Flanders is the northern part of Belgium. We have in this project funded by the Flemish Organization for Scientific Research 4 full partners, who are VITO. We are leading this project. And then we have the University of Antwerp with contributions on from the Toxicological Center and the Department of Critical Medicine. We at VITO, we contribute mainly with our environment and health team and the analytical chemistry team. Then we have the Ghent University. They contribute with a lot of experience and research infrastructure in the area of mycotoxicology and public health, and also a laboratory of toxicology where they, for example, do a lot of also clinical research with hundreds and thousands of samples. So they contribute, for example, to sample preparation and automation of that procedure. And then we have a third party looking over our shoulders. It's from the Flemish Department of the Environment, very closely linked to the funding agency in Flanders. And then I missed the KU Leuven, that's the University of Leuven, there in the middle, and that's Lode Goderis with the Center for Environment and Health. So what is the context of exposome research in Flanders? On the left, we have multiple stressors going from environmental stresses, non-chemical, like noise, lack of green space, but also the chemical substances like under REACH, or the pesticides or the biocides or food additives, and they often occur also in mixtures. We have the chemicals dependent on growth of fungi, mycotoxins, and then with several biological pathways, they might result in health effects like cancer, COPD, asthma, etc. And because Flanders is a densely populated, highly industrialized region, these pathways really are of interest to look into much more deeply. And then at the third, you always have the influence of lifestyle and socioeconomic variables. So it's not a one-way, one cause, one health relation. It's a multiple relationship issue. And in the middle, we can measure biomarkers of exposure and effect. Of course, you probably know all of this. And from IRENE Flanders research infrastructure, we try to contribute with large-scale longitudinal population cohorts with high throughput measurements of human and environmental samples, very similar to what we heard in the past 1.5 hours from RECETOX, obviously, and a data infrastructure to acquire and analyze high-dimensional data. And this all in an integrated and scalable approach with efficient workflows. In that respect, there's not a lot of new things, as I think a lot of this is very similar to what is being done in the Czech Republic. So as we tend to be now the Flemish hub, and in the future, very likely the national hub of IRENE research infrastructures, important is a cross collaboration between the different domains linked with health. So it's the environmental health on the left and the public health on the right, very similar to what I explained in my previous slide. And for this, we want to have IRENE Flanders with the research infrastructure in the middle. And overall, of course, we want to be a part of the European-wide research infrastructure, so distributed research infrastructure in the exposome field. So why do we want to have an IRENE Flanders? Why do we have an IRENE Flanders? Well, we want to strengthen the Flemish exposome research capacity. That's also what our funding agency wanted. They want to provide a scientific basis to respond to the risks of exposure to environmental stresses, but they also want to inventorize the potential links between exposure and environmental stresses, obviously. So then we talk about hypothesis finding, which can then turn into hypothesis testing. But for all of this, we need to increase scientific excellence, not only in Flanders, not only in Belgium, but throughout the whole of Europe. And that is why we are very happy to be part of the EIRENE PPP consortium, because in the end, it's our mission. I think it's the mission of all of us to reduce the environmental burden of disease. So just in a nutshell, what is EIRENE Flanders doing, and in what areas did we already invest in new infrastructures? Well, that's on the left, of course, the science policy interaction, where policymakers are largely involved in recruitment and sampling. We have a system from KU Leuven. On this, we are working with microsampling methods, and in the middle, we have the exposome toolbox that contain primary personal data with the results of the online questionnaires, the platform itself, but also wearable sensors and apps. Then below that, we have high throughput analysis, including high throughput sample preparation, and also proteomics with the Olink assay. And on the right, we have the personal exposome data hub. I will not go into detail into that because Eva, my colleague, will dive a bit deeper into that. And obviously, we are going from the left, from the physical research infrastructure, to on the right, more virtual research infrastructure with external data sources and external users on the bottom. Then just a snapshot. This is a result of work that has been done in the EIRENE PPP project by the work package that is looking at services that are already available throughout Europe. And they have categorized this into 10 categories. And I just wanted to give you an idea on which of these 10 that Flanders is contributing and will contribute even more in the future. That's the number 3, measurements of exogenous substances, very much links to mass spectrometry, targeted screening, but also untargeted screening. Then the 4th category, that's the omics-based analysis, where we will focus mainly on the proteomics part of the whole omics. Then we have the 6th category, it's the tools for biostatistics and bioinformatics, including platforms to investigate the exposome and the potential correlations and in the end associations and maybe causal relations with human health. And then the 8th one, it's the fair cataloging of exposome data. We have heard about this already also earlier today. So what is EIRENE Flanders research infrastructure contributing to already? Obviously to PARC, the second one, but the first one, it's a Flemish project, a Flemish program on exposome oriented research. It's already running. The first one was already in the early 20s. And now we are in the 5th cycle. Then in the middle, we have from Ghent, the Ghent Longitudinal Observational Research Investigating Aging, the GLORIA. It's a large-scale prospective cohort study focusing on age-related health outcomes. We have human sentinel studies organized by KU Leuven, where they cooperate with the occupational health physicians and focusing also on potential occupational exposure. Then we have Inquire, a large Horizon Europe project focusing on indoor air exposure. And then we have FlexiGut, which is investigating the impact of dietary and environmental exposure on chronic low-grade gut inflammation. I cannot see the time actually right now, so I think I will skip this one. We might come back later. So what is EIRENE Flanders trying to do? Of course, we are obviously, and several of our consortium partners, we are a cornerstone of the EIRENE PPP, and we are also contributing to PARC. In the past, we have contributed to HBM4EU. And of course, we are now trying to collaborate much more with other research infrastructures in Europe. And we will start, of course, in Belgium. Some of them you have heard already before, like Actris, BBMRI, but also ANAE on ecosystem services, and SHARE, which is also an infrastructure but then focusing mainly on aging in the population. Obviously, there's also some research infrastructures where Belgium currently at least is not contributing, like EIRENE on clinical research. And we are looking for new domains for partnerships in the domains of microbiome, personalized medicine, food, waste, product emissions, and sustainable chemistry. And with this, I would like to thank you, and I'm happy to answer any questions in the chat or orally. Thank you.
Thanks, Jos. Thanks very much. There are no questions in the chat, so we'll jump straight to Eva's presentation, which is on
Okay. I will share
HBM
my
data
screen.
services. Yes, please, Eva,
Yes.
go ahead.
I think you can see the screen now. Huh?
Exactly.
Yep.
Just put it into presentation mode.
Yeah.
Perfect. Yeah.
Okay. Okay, so I'm a colleague of yours, Eva Govers from VITO, and I will dive a bit into the VITO HBM data service, so the human biomonitoring data service that we are developing. So first, before diving into the content, let's situate a bit where these services are coming from. So within HBM for Europe, coordinated by the German Environment Agency, we were leading the work package on data management and data analysis. So that's why we initiated, as part of this work package leading role, to develop services for human biomonitoring studies to perform their data managements, etc. So it initiated the work in HBM for Europe. It's now further being maintained within the PARC project, and also the potential integration and the maintenance and the further elaboration will be discussed as part of EIRENE Flanders and EIRENE Europe. So the HBM data services, they're all integrated in a central platform. So we have a website developed where all these different parts can be found back. So currently we are hosting the European Human Biomonitoring Dashboard. We are hosting several tools to perform data management. And we have also integrated the platform to share individual level data for human biomonitoring data sets. And I will go more into detail in the next slides. So for the data owners to perform their data management, we realized within HBM for Europe that there was a lot of heterogeneity across the countries, how they performed the data management, for example, dealing with values below the limit of detection and the limit of quantification. So that's why we initiated a data management process to support all the data owners in the different countries to perform their data management. In the first step, we provided them with data templates and code books, how to harmonize their data, also specifically for the biomarker data. There was a biomarker list developed with unique codes for each of the biomarkers that could be analyzed in human samples. So here we provided them guidance, code books, biomarker lists, data templates to put their individual level data in the right format. In a second step, we also provided a tool for them to validate these data. So we can ask, for example, a data owner to provide a specific variable in 5 categories, but if they enter a 6 instead of a 5, we check this using the data validation tool. So all these tools are being performed by the data owner, him or herself, on its own computer, so they don't share the data with us, they perform all the steps themselves. Then in the next step, in the third step, we have a tool developed, it's almost released by now, to calculate additional variables, so to enrich the human biomonitoring data set. For example, we calculate variables where the values below the limit of detection, the limit of quantification are imputed. We also perform standardization creatinine, normalization for specific gravity, etc. So really we enlarge the data set in a uniform way. So for all the different studies, it's done on a central way, in the same way, to make this more harmonized across countries. So then they end up with a validated, enriched, large human biomonitoring data set, and then it's up to the data owners to decide if they want to share their individual level data with the PEH data platform. I will come back to that later. And also we have developed a collaboration with IPChem, the information platform for chemical monitoring. So there also they could share a subset of their data, so not the whole extended list of variables that they have gathered, but really focusing on the occurrence data that they have available And then we filtered this part out of their data sets. And after signing, of course, an agreement with IBCAM, those data could be shared with IBCAM. So this upper part is all on the individual level data. The down part is then we developed another tool to calculate summary statistics in a uniform way. So the different percentiles, the mean, the medians, etc. So that's also calculated in a standardized way. And then they have the possibility to share these aggregated data from their studies also with IBCAM and also with our European Human Biomonitoring dashboard. And I will come back to those two platforms in the next slides. So let's start with the European Human Biomonitoring dashboard. So we noticed when performing human biomonitoring studies, and also if you want to make publications out of it and you want to compare your data with other studies, that you really have to dive into the literature to find other study data. So that's why we decided within our HBM for Europe already to come up with a dashboard where data owners could share their summary statistics of their studies, because it's aggregated data, it can be considered anonymous, and so it can be shared publicly. So if you go to the dashboard, in the first step, you need to, of course, select which substance group, which compounds you are interested in, also the matrix and the unit. And if you if you click then on the apply selection box, you will get into the results of the search that you performed. So the results are structured in 3 different tabs. So in the first step, the exposure tab, the exposure data, so the biomarker data, are visualized using distribution plots. So the different selected percentiles are represented in the different plots over here. So here you can see for different studies from those sampling periods for the biomarker before how the distribution is organized, and also the LOD, the limit of detection, and the limit of quantification are included on the plots. Also, we have a lot of filters on top of the dashboard where you could select for specific countries, select for specific sampling period, also select for children versus teenagers, etc. So it provides a lot of flexibility to really search for the data that you are looking for. Then there is an additional tab, the second tab, the time patterns tab. So here the data are visualized by dots, a dot for the or the P50 or the P95. You can choose on top. And here the data are plotted according to the sampling period. So here it allows you a bit to visualize if there is a specific time pattern observed for the specific biomarkers. So here in this case for before, it seems that there is a small decrease over time. But of course, and that's also mentioned, I think, in the disclaimer, you always have to be cautious because you are combining and looking at different studies from different countries with different setups, not always general population probably. So you also have to be careful in interpreting those results. In the last step of the dashboard, you look into the health risk. So what do we do here? We compare the human biomonitoring data with available guidance values. So here again, in the first plot, the different percentiles are presented more in a simplified way. And then with the red line, the guidance value is given to give you an indication of how many participants have values above this established guidance value. And in the second plot, we calculate then the hazard quotient, so dividing the P50 and or the P95 over the available guidance value. And then you can see here the risk, the hazard quotient for the different studies included in the dashboard. So this really provides some supporting material for policymakers, for researchers, for risk assessors, etc. Then another feature of our website is the personal exposure and health data platform. So here really individual level data are stored, of course, after signing a protocol agreement. So the idea of our platform is to share this individual level data within projects. So it was established within HBM for Europe to be used during HBM for Europe, but also to make the data further available after HBM for Europe so that they don't disappear in all the data owners' computers, but that they are being used further. So you can submit a request to get access to the data, and we are basing ourselves on public interest as a legal basis to perform this activity. And also we foresee a secure environment, of course, because we are dealing here with pseudonymized data, to upload and download the data. So the data that are currently available in the platform are the data from the HPM for Europe Alliance studies and also the HPM for Europe MOM study. And later on, we will also host the data for the PARC Alliance studies and the occupation studies. Moreover, also with EIRENE, they are involved in this as well. And but there it's still under negotiation how to make the data available for after the project. So this is all handled by a really detailed procedure for data access. So if a specific partner within the project or outside the project wants to have access to the data, he or she first needs to submit a data access request form. This is evaluated by a committee. So the committee looks into the research objective, also if the data analysis plan makes sense. Also that only the variables are requested that are needed for the research objective, so the data minimization principle, the time frame of the proposal, and if there is not an embargo period on the data. After the DRAC has approved the request, then also again the supplying data controllers, so all the data owners from the different studies, get again the chance to approve or disapprove the request. And if everybody is approving, then the protocol agreement needs to be signed and access will be provided to the data. Another aspect with which we are working on, but that it's still work in progress, which is not yet integrated in the website, is the HBM metadata. So to get really from all the human biomonitoring studies all over Europe, an overview of the metadata that's available. So we did a lot of workshops, so metadata for machine workshops to get an idea of the content of the metadata that should be collected. That content step is more or less finalized. And now we are working more on the technical implementation to make a user-friendly interface to collect the metadata from those different studies. And also we are here in collaboration, of course, with the GRC team, with IPCAM. They're also involved in all these workshops, that later on, when we are gathering the metadata, the extended metadata in a central place, that a part of the metadata that is visualized in IPCAM, that it's being extracted from the big list of metadata. So to end with a last slide, so future steps and perspectives. So we are still finalizing 2 of the tools, the derived variables and the summary statistics. We hope to have it launched within a couple of weeks from now, so at least before Christmas holidays. Then we will also expand the dashboard. So for now, the only chemicals included in the dashboard are the HPM for Europe and the PARC priority chemicals, but we will extend it even more to all biomarkers that are measured in human biomonitoring studies. Also, we will extend our health tab with including all available health-based guidance values, and we have a collaboration established there with the Canadian dashboard developed by the ICES Human Biomonitoring team. Also, as mentioned, we are working on the metadata interface. We are definitely working also in collaboration, of course, and led, of course, by PARC W7 is the fairness level. So we are working on the semantics, the ontologies, reference vocabularies, also to make everything searchable, machine readable, catalogs of variables, and we will also, of course, connect with other data platforms. As part of the PARC Data Hub, we will also make sure that everything what we are gathering in our websites is findable and gets accessible through the PARC Data Hub. We, of course, are aware of the common data platform for chemicals, and we will definitely connect with them, specifically also to EEA, because they will be the responsible group for the human biomonitoring data. So we're well in touch with them and getting informed. And of course, we consider the HPM services that we developed as a valuable resource for various users, so including policymakers, researchers, and stakeholders in the public health and the environmental risk assessment domain. And here I can end the presentation and look forward to your questions.
Yeah. Thanks, Eva. Thanks a lot. Jana, go ahead with your question.
Thanks very much, Eva. It very nicely showed both your presentation and yours presentation, how we are developing the national nodes of EIRENE in parallel, but that they are also very complementary. And I think we are reaching the moment where we should really bilaterally start talking about how to link the things together, because we are not only showing our national infrastructures, but also how much we are already doing at the European level. We put more effort so far in the laboratories, aligning our laboratories, starting the small working groups, standardizing the labs. But we haven't started the data group within EIRENE, because a lot of it is happening in PARC. But now I see that we really have to start it, because, for example, what you presented about your HBM dashboard is very nicely fitted with what Richard presented about the Genesis, because we also have the visualization tools for air and soil and everything. Plus, we can offer that these data can be brought to the global reporting for the international connection reporting for conventions. So it means that we should probably set up a meeting, two of us, Rich, maybe a few other people, and really discuss in more detail how we bring together things that are already existing. And it can be also used towards the SIRENE project that we will be preparing the next year, because right now we are discussing different work packages and case studies that can be used. And I think this linkage of yours and ours resources would be very nice, and it could help PARC too. And it also explains the discussion that we have at the management board on Monday. I stressed out a couple of times that we are looking for the sustainable solutions. So in PARC and HBM, you did a great job in focusing on exposure and effect data. But from the bigger perspective of EIRENE, we also need to bring together all the multi-omic data. So we are talking about how we will link this to the genomic data, proteomic data, metabolomic data, non-target screening. And we can't really leave the data where they are and work with them remotely. And also none of our institutes want to develop the huge data storage, places for the raw sequencing data or something. And this explains why we set up the collaboration with EMBLI and EBI. EBI is interested in being, they can't be direct partner because they are not a country, but associate partner of EIRENE, and they are offering their services. And because they already have the software that is showing all the metadata from the cohort. Actually, that's the primary source software, and Molgen is a spin of it. And they have this information about the metadata available from the cohorts, also linked to where the data have, where the cohorts stored the genomic and proteomic and metabolomic data. This is why in EIRENE we are considering that we will develop in PARC the metadata standards for non-target data, for example, and they are offering to store it in their capacities. The same can be done for the metabolomics and sequencing. So this is why I said that we probably need to start this discussion and make some good and strategic and sustainable plan for the future, rather than making quick decisions. Something will go here this year and somewhere else next year. But this is great, and it really shows that we are moving forward individually, and now it's time to start bringing together, because in EIRENE Europe, we focused much more now. We focused on the building the pillars and inventorizing the services and building the political support. So little by little, we are building the house. But at the same time, all of us are working on separate rooms and national nodes. So maybe we can set up this bilateral meeting and discuss what we can already do.
I definitely agree. And I think it's also good that everybody made progress in their domains, but that now I think it's about time to also connect those domains. So really, definitely agree.
Very good. And thanks really everybody, because you've done the fantastic job with so many presenters. We really made it within 2 hours.
There's one question, Eva, to you, but Liz already dealt with that regarding time span from request to approval, and Adrian is thanking for the interesting presentation. So Hopefully that was useful for all of you. Thanks a lot for your participation. Unless anyone else wants to add anything, I would then, with thanks, stop this conference, and thanks a lot, and see you in other fora in the future. Thanks a lot.
Thank you too.
Thanks very much.
Bye-bye.
Have a nice
Bye-bye.
day.
Thank you for organizing this. Excellent.
Bye.